Yeah. This afternoon up here, but I mean, I had to take the question a couple times. I literally checked like every time for the last 96 hours.
Me too. That's what I'm saying. I haven't seen it.
But what I noticed was on like the main page, the date page, when I went to the grade sheet page, and I had to question there, and then I clicked on the link to the grade sheet, and then I had to click the question there.
Yeah.
But now I know they are. Do you want it? I have it on class.
Yeah, sure.
You excited?
No, but I need to
You did have.
I'm the secret. I'm the quiet one.
Right? What? How do you know?
I'm like the
Because I
ninja.
had a professor tell me that once. We were like all late. She said you're gonna do on the fifth grade, right? Old lady.
Look at my face. Don't forget the pictures, Nathan. 5.
I don't.
Yeah.
I got contaminated.
She's my secret. But it goes by alphabetical order.
Yeah.
But now you know.
I'm like,
But it's still, if you haven't done gram stains from it, we can do gram stains tonight.
Okay. Yeah.
Yeah, I'm not. That's not.
Yeah, it's just gonna be fun. Not a big deal.
I thought you turned in your other place. I did, but I mean, like, that's the one.
So all I did was extract DNA and then I put them back.
Oh, so this is the one you put back.
Yes. Now, and what they have on them now is a project number.
Yeah, I
You
saw
had
that.
project, and then I put a number on it.
Okay.
And then anytime you read it, make sure that number stays with it. Always keep that number.
Okay, so instead of writing Rosie's car, put Brian.
I'd be like,
You can put Rosie's car for one thing, but keep that project number because those things stay the same through the semester.
Okay,
It's
perfect.
a little contaminated too. Look at it.
Okay, so some of you may be really eager to have your exams back, but the exams don't come back until lab. So what we're gonna be doing now is starting on our next lecture topic. One of the things that I noticed in the exam, there was that section on different types of organic compounds, and quite a number of people were mixing up amino acids, proteins, nucleic acids, nucleotides. Once you get the exam back, and once I get a key posted, you have 1/4 of the final in your hands, and I highly recommend you go back and look at that exam and make sure you really know the organic compound section, because we're going to get into chemistry and we're going to be spending a lot of time talking about nucleic acids and proteins and metabolism and so forth. So it's a good idea to review. It's a good idea to review those exams and quizzes anyway, at least now and then, before we get to the end of the semester. Okay, so last time we were talking about microbial nutrition and growth, and that kind of went along with the exercise that we did involving microbial nutrition and growth, which we'll look at the results for that tonight. Now we're going to get into the section on metabolism, and along with this there's some terminology. We're going to talk about enzymes, ribozymes, bioenergetics, metabolism. So we'll get started on that. Notice it says enzymes, ATP, and bioenergetics, but not in that order. Well, so what is bioenergetics? Anybody know? Energy can't be produced,
Energy.
contrary to the first law of thermodynamics. Wait, what?
Energy transfer within living organisms.
Energy transfer mechanism, and it's very, very significant that you remember, put into your brain and don't let it get kicked out. Living organisms cannot produce energy, because there are lots of references that will tell you they do, and they don't. We don't. Living organisms can't. Energy can't be created. That's what the first law of thermodynamics says. Energy can't be created, it can't be destroyed. It can, however, change form. And so transfer mechanisms are going to happen, and that's what bioenergetics is about. This is a kind of foreign term. Not all textbooks talk about bioenergetics, but it's really important. So it is energy transfer mechanisms occurring within living organisms. So it turns out this is very closely tied to metabolism. We'll see why in just a little bit. But why is energy transfer significant? Why does energy transfer have to happen? To think about that, think about what are the energy sources of living organisms. Well, as I said, energy can't be created, it can't be destroyed, but where does energy come from then? If organisms have to have it, they can't produce it, they can only transfer it, they can get it from external sources. What are the external sources? Think about our nutritional categories. What are the sources of energy?
Organic compounds.
No, that's just chemicals. That had to do with carbon. Energy sources are chemicals and light. Our energy sources. Again, think about our nutritional categories. Our energy sources are chemicals and light. This is talking about ATP. And ATP is a high energy compound that organisms use to run their metabolic processes. But ATP isn't stored in the cell in large quantities, so cells are constantly having to make it, which means they're going to have to get energy from some external source and then transfer that energy to ADP to make ATP, a high energy compound. Well, so again, the sources, light and chemicals. Now I know that heat energy does cause molecules to move, but I've never seen anything in a textbook that says heat energy can be used to run metabolism. It's one of those things that I wonder about, because we do have organisms that live on the seafloor next to the thermal vents. There's a lot of heat energy there available. As far as I know, they can't capture that directly and use it to make ATP. Or if they do, I've never seen anything written about it. Maybe some archaea can do that, I don't know. Relative to the organisms I'm familiar with, their sources of energy are either light or chemicals. Again, think about our nutritional categories. If they use light, they're phototrophs. If they use chemicals, they're chemotrophs. And then chemoauto or chemohetero is where you get into inorganic or organic. Okay, we say that energy flows through living systems, and this is because it can't be created, it can't be destroyed, and also because some of it gets lost. Energy is coming from external sources, light or chemicals. That energy is going to be used to make ATP, but the processors are not 100% efficient. It's like, what do they call those? Perpetual motion machines. They don't actually exist, because to get things running, to keep things moving, a certain amount of energy gets lost. Entropy comes into play. So entropy is this tendency toward disorder. A certain amount of energy is constantly being lost. And in the diagrams, you can see an ordered set of ice crystals that are coming apart. When the ice crystal melts, that might be a snowflake. And then in another diagram, you see a highly ordered particle breaking up into a bunch of smaller particles. Well, so entropy is just always there. A certain amount of energy is going to be lost due to entropy. And this is why living organisms constantly have to have an external energy source to run their metabolic processes. They're constantly transferring energy, and some energy gets away due to entropy. So they have to have an energy source, always building ATP. So we did say that maintaining a high degree of organization was a characteristic of life, and maintaining that high degree of organization, think about all the different types of organic compounds, carbohydrates, proteins, lipids, nucleic acids. Building those organic compounds, that's organizing, that requires energy. So they're having to have an energy source. Now, what is metabolism? That's a term you should definitely know.
Breakdown and building.
But just in general. So you can say breakdown and build up. Think of defining terms with full sentences. When I'm reading definitions, I'm seeing a lot of people put little points, but it's like, okay, you've got the little points, but put the whole thing together in a big picture. What is metabolism? It's all the chemical reactions occurring within living organisms. All the chemical reactions. Yes, some of them are breakdown reactions, some of them are building reactions. So we had two terms when we looked at metabolism. Metabolism, all the chemical reactions. And again, it's not chemical reactions happening in the chemistry lab. It's chemical reactions taking place within living organisms. It's happening in association with protoplasm. The two general categories are, taking things apart is catabolism, building things is anabolism. So we're going to tie this to energy transfer. Anabolic reactions. Anabolism is building, and building reactions, putting small particles together, increasing organization, that requires energy. Anytime organisms are building big molecules, that's going to require energy. So reactions that require energy are said to be endergonic. If in the process you would notice a change in temperature, it's endothermic. But for now, let's not worry about the temperature. Endergonic just means they require energy. And this energy is typically going to come from ATP, because ATP is the energy currency that cells are using. So the cells can take apart ATP. They're going to release the energy from ATP, and it's going to become ADP and phosphate, and the energy is going to be used to build something within the cell. So in this case, an anabolic reaction, our energy is coming from ATP. That's what this represents. ATP is going to be used to put these particles together and build this complex compound. It's going to actually have to take place in a series of steps. But building reactions, anabolic reactions, are endergonic. They require energy. Meanwhile, breakdown reactions, which are called catabolic reactions release energy. I have to qualify that because they're going to release more energy than was required to get them started. Typically, there has to be some activation energy, but once the reaction is going, there's going to be more energy released. So they are exergonic. Again, if there's heat released, it's exothermic. But for us, exergonic reactions release energy more than what's required to get them started. Basically, what's happening when organic compounds are taken apart, covalent bonds are being broken. And when those bonds are broken, energy is released. Remember, this is a class where you're supposed to be paying attention to me, not your phone. Especially not if it engages this guy over here, too. That's called majorly distracting. Please don't do it. It's very rude. So again, external energy sources required. Organisms have to have energy either from light or from chemicals, and they're going to use that energy to form ATP, and then use ATP to drive their metabolic processes. We're going to spend quite a bit of time talking about this. Again, organisms don't store ATP in large quantity. It's not something that they can have a big stack of or a big pile of somewhere within the cell. It's something that they make, and it's used up. They make it, they use it, they make it, they use it. It's constantly turning over. So you'll see this kind of a diagram. ATP is what's being made. It's being shown as a high energy compound. It's made by binding ADP with inorganic phosphate. P with a little I behind it is inorganic phosphate. So you have to have energy to put these two together to form ATP, and when it comes apart, energy is released. This is going on all the time. From a molecular standpoint, and I'm giving you this acronym, ATP, what does it stand for? Adenosine triphosphate. So adenosine triphosphate, this is referred to as a high energy compound. It's referred to as a nucleoside triphosphate. Biology textbooks tend to call them nucleotide triphosphates, which is not right, because a nucleotide is a sugar, a phosphate, and one base. If you've already got one phosphate on there and you put on 3 more, you'd have 4, which ATP isn't. So it's a nucleoside triphosphate. Anyway, 2 phosphate groups are added to a single nucleotide, and the base for that nucleotide is adenine. Typically, ATP molecules include the pentose monosaccharide ribose. So when you're talking about ATP that the cell is using on a regular basis, usually the sugar is ribose. As soon as I say that, later on we're going to be talking about making making DNA. If a cell is making DNA, then the sugar has to be deoxyribose. So there's 2 different forms of ATP. There's ATP with ribose as the sugar. There's ATP as with deoxyribose. But this is ribose, and one of the characteristics we see is there's 2 hydroxyl groups down here. This is the adenine. These are the 3 phosphates that are on there. So adenosine triphosphate. Energy is going to be released when the covalent bonds between the phosphates are broken. And there's a little bit of energy in the first bond binding the phosphate to ribose, more energy that's binding the first two phosphates together, and then a very large amount of energy binding that third phosphate on there. Sometimes that last set of two phosphates is called pyrophosphate, but what's going to happen is ATP is going to be hydrolyzed. So we talked about hydrolysis reactions, where water is added, ATP comes apart, and you get ADP, and this is the inorganic phosphate. So again, PI is this inorganic phosphate, and energy is released, 7.3 kilocalories per mole. Good news for you guys is we are not going to be calculating any kind of stoichiometry, never mind. Yes, you can spend a lot of time doing that. We won't. Okay, so we think of ATP as energy currency. Again, high energy compound, the most important high energy compound within cells. It's being formed, it's being used on a regular basis. We're going to spend quite a bit of time talking about how it's made. There it is, high energy. To give you an idea of what the turnover is, supposedly a typical eukaryotic cell can use as many as 1 million ATP molecules per second. As soon as I say that, I think, what is an average eukaryotic cell? Are we talking a neuron? Are we talking an amoeba? Planet-Monas? I have no idea. I don't know what an average eukaryotic cell is. Still, 1 million ATP molecules per second. That tells you metabolism is happening pretty rapidly. And again, if they can't store it, they must constantly be making it, which they are. Cells are constantly making this stuff. They have to. And again, the energy that is going to be released when ATP is broken is stored in the covalent bonds between the phosphate groups, especially between the second and third phosphate. That's the pyrophosphate bond. And when it's broken, energy is released to do work in the cell. So you'll see these diagrams, ATP going to ADP plus phosphate. In that case, the I is missing off the phosphate, it should be on there. And then we think about what is some of the energy or some of the cellular processes we talked about that require energy. Moving a flagellum. Whether it's eukaryotic cell or prokaryotic cell, moving a flagellum requires energy. Actually, prokaryotic flagella probably use a hydrogen proton flow to spin, but to get hydrogens on one side of the membrane probably required ATP in the first place. Active transport requires ATP. Endocytosis, exocytosis, walking of kinesin, dynein, those things require ATP. Plus all the anabolic reactions that are happening. So this again is just showing you a chemical reaction, very simplistic, where energy from catabolism of some organic compound, this would be something a chemotroph would do, where they're breaking some type of organic compound, they're releasing energy, that energy is used to convert ADP and inorganic phosphate to ATP, and then ATP can be broken down and energy can be released. This is showing the same thing, except that notice here it says the energy could come from either chemicals or sunlight. And that's what I said, your external energy source has to either be chemicals or light. And again, PI represents inorganic phosphate, and you might just see that as P, or you might see it as PO4. Now, in total, chemical reactions that yield ATP, so if a cell is making ATP, the chemical reactions involved are called phosphorylation reactions. All processes involved in making ATP are phosphorylation reactions, but not all phosphorylation reactions yield ATP. Hopefully that won't get confusing. Phosphorylation reactions can be divided into categories, and we're going to talk about these. They're substrate level, oxidative, and photophosphorylation. Just looking at those terms and thinking about things we've talked about earlier, how do these differ? Phosphorylation reactions, they all have to do with making ATP. If it's photophosphorylation, what's the energy source? Light. The other two, substrate level phosphorylation and oxidative phosphorylation, the energy is coming from chemicals. So one of those is energy coming from light. The other two are energy coming from chemicals. But we're gonna see some significant differences between these. The easiest one to think about is substrate level phosphorylation. And a substrate, the term substrate typically refers to something that a cell can take apart. Sometimes those substances are called catabolites. But a substrate, if you're thinking about chemical reactions, the substrate is something that a catalyst can work on, and the substrate is going to be taken apart to release energy. If the energy is used directly, when you break a substrate, you break a covalent bond, and you use that energy directly to make ATP, that's called substrate level phosphorylation. In this particular example, you see the phosphate group coming off of some phosphorylated molecule here. Oftentimes it's carbohydrate, so it might be something like glucose 6-phosphate, except it's only got 3 boxes, but anyway. If this were a glucose 6-phosphate can actually transfer its phosphate to ADP to make ATP. That's substrate level phosphorylation, where the energy is coming directly from the substrate, which is being broken. Sometimes it happens that way, sometimes it doesn't. So it may not involve the transfer of a phosphate group from the substrate directly. But if the energy is coming more or less directly from the substrate molecule being taken apart, then it's substrate level phosphorylation. This is the simplest method of making ATP. The other two, oxidative phosphorylation and photophosphorylation, I've mentioned these terms in association with eukaryotic organelles that were membranous. Remember that I said had evolved from bacteria? Those membranous organelles were mitochondria and chloroplasts. Mitochondria do oxidative phosphorylation, chloroplasts do photophosphorylation. What's involved in that is the inner folded membrane of chloroplasts or mitochondria. That inner folded membrane, either thylakoids or the tristia of mitochondria, have these elaborate series of proteins associated with the membrane. Here is the last molecule in this diagram. This is ATP synthase. And ATP synthase is the molecule that's actually going to convert ADP plus phosphate into ATP. So this is very important. But we're going to be looking at some of these other molecules that are along this line. This is a cytochrome chain. Both oxidative phosphorylation and photophosphorylation involve something like that. Which as soon as I say that, I think, sometimes. So very often you'll have a cytochrome chain involved, and what they're doing is moving hydrogen protons to one side of the membrane. The hydrogen then comes back through ATP synthase, and that hydrogen flow, that hydrogen proton flow, is what actually provides the energy to make ATP. That's always the case, but how the protons got across the membrane in the first place is variable. Because remember bacteriorhodopsin? It just moves protons by itself. In that case, you don't have an electron transport chain. And if this is all totally foreign to you, don't worry too much. We'll get into it in excruciating detail. Okay, so ATP is the most common form of nucleoside triphosphate. ATP, because it's A, that has adenine. What are the other nitrogenous bases? Guanine, cytosine, thymine. So there are other nitrogenous bases, consequently there can be other types of nucleoside triphosphate. ATP is not the only one. So here we see the other nitrogenous bases, thymine, cytosine, adenine, guanine, or adenine we talked about. So any nucleotides that contain other nitrogenous bases can also become high energy compounds. In fact, they have to for making the nucleic acids, DNA and RNA. You have to have high energy compounds involving thymine, cytosine, uracil, guanine. So ATP is not the only one. You can have GTP, CTP, UTP, etc. And then there are some other high energy compounds that are sometimes incorporated into metabolic processes. So two that are fairly significant are acetyl coenzyme A and succinyl coenzyme A. Coenzyme A, well, coenzymes are enzyme helpers. I'll talk more about coenzymes in a little bit. Acetyl coenzyme A is a pretty complex looking molecule. So here you see you have these ring-formed compounds, you've got 3 phosphate groups, you've got this chain going out this way, so this is a pretty big molecule, but it is a high-energy compound, acetyl coenzyme. It can transfer energy to other compounds, and succinyl-CoA can do the same thing. I don't know if I left it in the lecture syllabus. Creatine phosphate, or phosphorylcreatine, is another high-energy compound, but it's associated specifically with human muscle cells, and this isn't human physiology, so never mind. But you know, we're not gonna talk. Okay. Now, metabolism. All those chemical reactions that are happening within living organisms, those chemical reactions are dependent upon substances called catalysts in order for the chemical reactions to take place fast enough. So catalysts are compounds, they're molecules, and their job is to speed up chemical reactions. They are not changed by those reactions. In other words, they're not part of the substrate, but they are going to be required in order for chemical reactions to take place fast enough to sustain life. So you have to have these catalysts. They're going to increase reaction rates, and it used to be thought maybe they increase reaction rates maybe tens of times or 100s of times. Now we know that they increase reaction rates thousands of times. They're highly significant. They're going to speed things up. They cannot cause chemical reactions to take place that wouldn't otherwise. In other words, it has to be something that could have occurred, the chemical reactions could have occurred. They're just going to make it happen faster. They're going to speed them up, and they're going to speed them up in a very big way. This is showing one type of catalyst. This is an enzyme, diagrammatic representation. So in this case, this enzyme has a specific configuration. This is called the active site, and it can bind with this substrate molecule. And then maybe take it apart. Is everybody in here? That door's locked. I suppose I should unlock it so if anybody wants in. Anybody know of another type of catalyst?
Enzymes.
So enzymes are catalysts. The alternate type is a ribozyme. A ribozyme is another form of catalyst. That's a ribozyme. It used to be thought that the only biochemical catalysts that were of any significance were enzymes. Now we know that's not the case because ribosomes are also catalysts. So how do they differ? Well, enzymes are proteins. Enzymes are always proteins. And proteins, we know, are long chains of smaller units. If they're enzymes, they're very likely to be globular in form. Sometimes they have quaternary structure, which means what? More than one polypeptide chain. So they're pretty complex. And what are those things they're made of? Amino acids. Proteins are long chains of amino acids. And the amino acids are held together by what kind of covalent bonds? Peptide bonds. Okay? So I made those blue because when I was talking about proteins a long time ago, I said proteins are going to be color-coded blue. And the individual units that make up proteins are amino acids. And amino acids are held together by peptide bonds. Consequently, if you have a long chain of amino acids, it's called a polypeptide, which may or may not be a complete protein because some proteins are quaternary. Hemoglobin, if you only have one polypeptide, you don't have a hemoglobin molecule because it has 4. So ribozymes are RNA molecules. They're generally smaller than enzymes, although enzymes can vary quite a bit in their size. Some of them are quite large and very complicated, others are not. But RNA molecules are made up of nucleotides, and the nucleotides are held together by phosphodiester bonds. Again, this is kind of a review of the chemistry stuff that we talked about, talking about composition of protoplasm. Those are really important to know. What are proteins? What are the building blocks of proteins? What are nucleic acids? What are the building blocks of nucleic acids? To recognize that both of these can be catalysts, speeding up chemical reactions. Again, these molecules are not changed by the reactions they catalyze. As soon as I say that, it's like, oh, okay, they might be changed a little bit, but they're not changed permanently. When I say they can change a little bit, if they're globular proteins, they have a kind of rolled up in a ball, the surface structure, the surface configuration can change a little bit and then go back to its native form after the reaction has been catalyzed. Because they're not changed significantly, they can be used over and over and over again. It doesn't take a lot of these molecules to accomplish metabolic processes because they're not themselves degraded in the process. On the other hand, they don't last forever. They will eventually break down. Especially RNA, there are molecules called RNases, and RNases degrade RNA, so they're constantly taking those things apart. Now, the other thing about this whole section we're getting into, so we're going to be talking about metabolism, it's not something that you can look at under the microscope. I can't show you metabolism happening. Instead, what you're going to see is diagrammatic representations of things, molecules, and what's going on. Sometimes I'll show you movies that show you what's going on. These are all models that have been put together to kind of give you an idea, in this case, how enzymes work. So there is one example or one model for how enzymes work. It's called the lock and key model. What this tells you is that each enzyme has a specific reaction that it will catalyze, just like each lock has a specific key that goes with it. At least we hope so, because often we have house keys or automobile keys, and it's kind of unsettling to think there might be 5,000 that can open the same door. We hope they're specific. So in this case, this is a really simple diagram. It's showing how these two substrate molecules, this blue one and this pink one, they can both be held by this enzyme, and then what the enzyme is going to do is cause them to be bound together. So it's going to speed up the reaction, pulling those together. They wouldn't fit with any other enzyme. They have very specific, this is called the active site of the enzyme, active site or reactive site, and it's very specific. Here it's shown as fitting that particular key, but here it's just a rectangular piece and a triangular piece. Anyway, substrate molecules are supposed to fit very specifically with their enzymes. BJ, if you're standing up, people can't see that are behind you. Blind. So each enzyme has an active or reactive site. It's supposed to fit specifically with a substrate molecule. Oftentimes more than one substrate molecule if they're putting things together. And substrate, another term for substrate is reactant. So what's going to react? They're either called substrate or they're called reactants. So here we see a couple of other examples where the enzyme has a specific site. So here's the reactive site. If this is the enzyme, this substrate is going to fit in there. Once it's in there, the enzyme can take it apart. That would be catabolism. If we were running this the other direction, these two particles can fit in here, they're going to get put together, and then a product is released. Enzymes do both. Sometimes the same enzyme can run a reaction either way, depending on the concentration of reactants or product. Now, as I said, this was kind of an old model, the lock and key model. It was visualized that these proteins had a specific reactive site that fit exactly with the substrate. More recent evidence suggests that instead, when substrates bind, they actually modify the enzyme just a little bit. This first diagram, it's really hard to see any change in the enzyme reactive site, but apparently, as the substrates bind, the configuration of the protein changes just a little bit on the surface. This is going to involve weak bonds, so maybe hydrogen bonds, maybe ionic bonds. Anyway, not covalent bonds, where they're sticking to the surface. But the enzyme is going to change its conformation just a little bit to fit the substrate. This next diagram shows it better because it looks like the enzyme has an open mouth. So here you can see the enzyme ready to pick up the substrate, and then when it gets it, it actually changes its conformation and binds with that substrate more permanently. This is referred to as the induced fit model. So it's as though you go to put your key in the lock, and the lock kind of changes a little bit in shape to fit your key more precisely. And they think that's a more accurate model for how enzymes work. So basically, again, what the catalysts are doing is speeding up chemical reactions. You have substrate molecules that could interact, but they rarely come together. Then the job of the enzyme is to speed up the rate at which they come together. And they can do that by binding both of them at slightly different positions and then putting them together so that a product can be formed. Again, it can be shown diagrammatically. And then when the product is released, the enzyme is free to be used again. I can give you another analogy that is a story analogy. Okay, so I can show you lots of diagrams, and you can go online and find lots of diagrams. But imagine instead a playground situation where you have two little boys that don't like each other. We'll call them Billy and Bobby. Billy and Bobby know they don't like each other, so they stay away from each other. Along comes Susie. Susie is the catalyst. And Susie goes to Bobby and she says, you know what Billy said about you? Blah, 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 blah. Then she goes to Billy and she says, you know what Bobby said about you? Blah, blah, blah. The next thing you know, Billy and Bobby are together. Susie, the catalyst, is not involved in the together, so she could catalyze that reaction over and over again. Unless the other guys got smart. But anyway, there's another story that involves camels, but I can't ever remember how it goes. Anyway, so it kind of gives you an idea how enzymes work. They just speed up chemical reactions, make them more likely to occur. They would have occurred anyway, or they could have occurred anyway. And again, both catabolic and anabolic reactions. Well, and the same is true for ribozymes as far as I know. So some enzymes take things apart, some enzymes put things together. Another kind of model for how enzymes work has to do with energy, the energy that's required for chemical reactions to take place. So we say that enzymes decrease activation energy. Well, they seem to increase the increments of activation energy because what they will cause to happen, instead of having one single step reaction, they'll cause reactions to happen in a whole series of little bitty steps where the activation energy can be very small. So I can give you a specific example of this. Glucose can potentially interact with oxygen in the air. And if it does, if glucose reacts with oxygen in the air, you can get carbon dioxide and water as waste products. That's chemically feasible. But if you have glucose in a spoon or sucrose in a spoon, what do you have to do to get that to react with oxygen in the air? You're going to have to put in a lot of energy. Another way to think about this is a marshmallow on a stick, because then you have a better idea of what to do with it. If that was glucose, and you wanted to get that glucose to react with the oxygen in the air, you have to put in energy of activation. You're going to have to get it hot. Well, we have Bunsen burners in here, so we just put it over the fire. It will ignite. And as it's interacting with oxygen in the air, carbon dioxide is going to be given off, water is going to be given off as vapor, and a lot of heat and light are going to be given off. And the sugar, some of it's going to form this gooey black mass in the bottom of the spoon. But is that conducive to life within cells? Not so much. I mean, you can visualize if you did that with a frog, he'd either get off the spoon or he'd be gone. So you don't do that. So heat's great activation energy for something like this, but it's not compatible with life processes. And anyway, we already said metabolism uses what for energy? It's not heat. What is it? But when we're talking metabolic processes where you're having a lot of chemical reactions taking place within the cell, what's the energy source? ATP. ATP provides energy in tiny little increments. So the energy is provided in little bitty bits. The diagram on the right here is showing the reaction without enzymes. So if we're going to have sugar react with oxygen, we're going to have to put in all this activation energy, a tremendous amount of heat. If we want this to happen within the cell at temperatures conducive to life, we can't do that. So we're going to have very tiny increments activation energy is going to be provided. This looks like, well, you're not getting nearly as much change here. And this is because this has to happen multiple times. You can have a whole series of chemical reactions to get oxygen to interact with sugar, because it doesn't happen directly. It's going to happen through a whole series of events. Guess what? You get to learn about those. So this is showing what has to happen if you were starting with table sugar, which is sucrose. The first thing that would have to happen is you have to take it apart. Sucrose is sucrose is a disaccharide. It's made up of glucose and fructose. So you have to take those apart. And then you can start taking the glucose apart. But you can have a whole series of events. How many of you have seen that? Yeah. So some of you have seen this before. This is the glycolysis pathway. This is the beginning of this situation. Remember when I was talking about fermentation associated with our food microbiology, and I had glucose and a little tiny arrow and then pyruvate? Well, here's where we start. This is glucose. Here's where we end. This is pyruvate. So that's only the first part of getting sugar to react with oxygen. We have a lot more to go here, but anyway, we're going to talk about glycolysis in quite a bit of detail. The activation energy provided here is ATP, and it's provided there and there. So energy has to be provided. Get this reaction, series of reactions going, little increments of activation energy, and in the process, we're going to get energy out. This is additional reactions that have to take place, because so far, we've only got the sugar to be taken apart to pyruvic acid. Then pyruvic acid is going to drop into the Krebs cycle, which is this thing, also called the citric acid cycle. There's some intermediate steps here, and we're going to get going in this cycle. We're going to pull carbons off. We're going to release a bunch of coenzymes in the reduced form. And then finally, we're going to run an electron transport chain, because we want to get our sugar to interact with oxygen. Only you know what? It doesn't directly. If you're talking about cells within our body, oxygen doesn't get involved in the process until way out here at the end. Here's oxygen picking up electrons from this electron transport chain, and the oxygen is converted to water. But this electron transport chain allows hydrogen protons to collect on one side of the membrane, and they come back through ATP synthase, and we make ATP. It's not like putting sugar in a spoon and igniting it. Instead, it's a whole series of chemical reactions. Glycolysis first, and an intermediate, then the Krebs cycle, then the electron transport chain. A bunch of little tiny reactions where the energy of activation is ATP. Or, in the Krebs cycle, acetyl coenzyme A, succinyl coenzyme A. Well, anyway. So, the point is, enzymes decrease the increments of activation energy, so that we're going to catabolize sugar, but we're going to do it in a whole series of tiny little steps. And the whole thing doesn't have to get hot. So again, energy is provided by ATP, acetyl-CoA, succinyl-CoA, some other molecules, but anyway. Why is this important? Well, why do cells break down glucose? So if you were putting glucose in a spoon and burning it, energy is going to be released. It's going to be released in the form of light and heat, maybe some noise associated with it. Again, that's not what cells want to do. Cells are going to break down glucose because they want the energy that's stored in it. Glucose is 6 carbons attached together. Every time you pull a carbon off, there's a potential release of energy, which can be used to make ATP. So when cells convert glucose to oxygen, they use 2 molecules of ATP, 2 succinyl-CoA, 2 acetyl-CoA, and in the end, they get out either 36 or 38 molecules of ATP. So they get a lot of energy out. There's some energy that gets away in the form of entropy, as I said. But you can't use heat. Not conducive to life. Now, we're going to talk some more about enzymes. There's more information here about enzymes than about ribozymes. I'll give you two specific examples of ribozymes, but we have a whole bunch of different enzymes that we talk about in here. So first of all, we can categorize enzymes. There's a lot of different ways to categorize them. Some of it has to do with their structure, what they work with, what they work on, how they're regulated. There's all kinds of things. So our first way to categorize enzymes is endo versus exo. What do you think those terms would mean? It has to do with where is the enzyme active. If it's an endoenzyme, it's active inside the cell. If it's an exoenzyme, it's active outside the cell. So then, why would cells go to all the trouble to build proteins, enzymes, and then dump them out? Don't look now, but our cells do this too. So why? Why do cells construct proteins? We're going to see it takes a lot of energy to do that. Building proteins, that's anabolism. Anabolism requires energy. Why do they do it? Digestion. Exactly. They dump enzymes into their environment. Fungi do this, bacteria do this, we do this. Dump enzymes into the environment that will break down organic compounds into smaller units that we can then take in. It's a lot easier to take in a glucose molecule than it is to take in a polysaccharide. So we put digestive enzymes out into the environment. So a lot of exoenzymes are digestive. Not necessarily all of them. Another way to categorize enzymes is simple versus conjugated. Simple enzymes are proteins alone. So they are just protein, they're active as protein. But if an enzyme is conjugated, that means it has to have a helper associated with it, and the helper is not a protein. It's some kind of a non-protein entity. There's some variation in terminology depending on whose book you look in, which can be confusing because it's inconsistent. So I'm going to use a terminology that I think is a bit more consistent. If you look at this diagram right here, notice this word, cofactor, non-protein portion, also sometimes called an activator, but it's called a cofactor here, and up here it's called a coenzyme. I'm going to tell you those two are not the same. Some references call all the helpers cofactors, and then say they're subcategories of cofactors. I'm going to just call them helpers, and then say there are subcategories of helpers, and cofactor is one. Anyway, so if you have a conjugated enzyme is one that requires a non-protein helper. If the non-protein helper is not with the enzyme, then the enzyme is inactive, and it's called an apoenzyme. An apoenzyme is the inactive form of a conjugated enzyme. It's protein alone, it can't do anything. When you put the helper with it, then you have what's called a holoenzyme. The holoenzyme is the active form of a conjugated enzyme, so it's protein plus non-protein helper. And the helper groups vary. So our first helper group, I'm going to call coenzyme. Coenzyme is a specific category of helper. Coenzymes are organic. They're non-protein, but they are organic compounds. They're pretty good size. So these are relatively large molecules. Again, what is a coenzyme going to do? It's a helper. It's going to bind with the protein apoenzyme and make it into a holoenzyme. So it's going to convert something that's inactive into something that's active. And you've been introduced to some coenzymes already because we said nucleotides could be used to build coenzymes. And then I gave you names like NAD, FAD, NADP. NAD is nicotinamide adenine dinucleotide. FAD is flavin adenine dinucleotide. That tells you, yes, they're made from nucleotides. However, they're also derived from vitamins. NAD from niacin, FAD from riboflavin. So why do we take vitamins? Well, why do you take B complex vitamins? Because they're used to make coenzymes, and you have to have coenzymes in order for your enzymes to work. If you have enzymes that are conjugated enzymes, they can't do anything without their non-protein helper. Some of those helpers are NAD and FAD. Now we're going to be looking at a rather selective function of these coenzymes because they have more functions. But again, this is an introductory class. You have to roll the chair. I just noticed you're still standing.
Yeah.
Sorry.
Thanks.
I keep forgetting to get your stool. Takes me a long time to notice, sorry. Anyway, so know that NAD and FAD are coenzymes. Know that NAD is derived from niacin. Our FAD is derived from riboflavin. Those are both B complex vitamins. And then we're going to see these things being used, and basically what they're going to do is pick up electrons and then donate electrons to other molecules. Without that, the enzymes they're bound to don't do anything. Now the term cofactor, some references use cofactor to describe all the helpers. I'm going to say cofactors are not coenzymes. Cofactors are inorganic. They tend to be minerals, so they're inorganic groups. Again, their function is to bind with an apoenzyme, which is inactive, and make it a holoenzyme, which is active. Some examples of cofactors include calcium, magnesium, manganese, even molybdenum. So the reason our azotobacter medium contains molybdic acid, because molybdenum is a cofactor. There's some enzyme in there that those organisms are using that requires that cofactor. Probably has to do with nitrogen fixing. Anyway, so if they're cofactors, they're inorganic. If they're coenzymes, they're organic. And then there's one more category. There are minerals that can serve as prosthetic groups. Prosthetic groups are inorganic, but they're permanently bound, so they don't come loose. Prosthetic groups typically are iron, copper, zinc, some type of metal. And the enzymes that hold those are sometimes called metalloenzymes. Here's an example. This is heme. Heme is a group that's associated with enzymes called cytochromes. The cytochromes have iron prosthetic groups. As soon as I say that, I think some of them have copper. But the iron in this case, in the heme, the iron is right here in the middle. This heme is also associated with hemoglobin, which isn't an enzyme, but hemoglobin has polypeptide chains coming off of these corners, and that iron is part of that heme group. So it is a prosthetic group. It's permanently bound. It's not going to come off. And a group of enzymes called cytochromes, the term means literally cell color, because these things are pigmented enzymes. Most of them have iron prosthetic groups. Sometimes not. I believe there's cytochromes that have copper prosthetic groups. We usually think of them as having iron. And they're going to play a major role in oxidative phosphorylation and photophosphorylation. At least photophosphorylation as it occurs in algae and cyanobacteria, but not photophosphorylation involving Bacteroidetes, which is in the archaea. So again, these helpers can serve a variety of functions. Coenzymes, cofactors, prosthetic groups. They can have more than one function. What we're going to pay attention to is their function as electron donors and electron receptors. They pick up electrons, they donate electrons, so they're involved in oxidation and reduction reactions. How many of you are familiar with oxidation reduction? Okay, so again, from a chemical standpoint, there's calculations that have to be done about oxidation potentials, reduction potentials. We don't do that. We're not doing calculations, but you do need to know what's oxidation and what's reduction. The other thing about these helpers, the enzymes are very specific in terms of what they can do. Think of the lock and key model. Each different chemical reaction that has to take place within a cell has to have a different enzyme or a different ribozyme. But the helpers are not nearly as specific. So you'll see the same coenzyme used for a whole bunch of different enzymes. So they can be involved with more than one enzyme. And sometimes they're called co-substrates, but I don't like that term, so I won't call them that. Okay, so we're going to see our coenzymes and we're gonna see sometimes cofactors, but most often the coenzymes, NAD, FAD, we're gonna see them involved in oxidation reduction reactions. So if you're familiar with them, great. If you're not, we're gonna review oxidation and reduction real quickly. So oxidation reactions, those are reactions that involve the addition of oxygen to, or the removal of electrons and hydrogen protons from atoms or molecules. When you think of something being oxidized, think of iron being oxidized. When it does, it forms iron oxide, which is rust. But when you're dealing with living organisms, it turns out that oxidation reactions more often involve that removal of electrons and hydrogen protons. So we're going to see a lot of different compounds get oxidized. When they're oxidized, they give up electrons and protons rather than gaining oxygen. There's one example, we'll talk about beta oxidation. Beta oxidation, that actually is an oxygen attached, but usually it's loss of electrons and hydrogen. Reduction reactions involve the removal of oxygen from or the addition of electrons and hydrogen protons. This can get confusing if you think of reduction as something getting smaller, because in actuality, when molecules are undergoing reduction reactions, they don't get smaller. They pick up electrons and hydrogen protons, so they actually get bigger, not significantly bigger, but they do. So another way to think about it, if reduction involves addition of electrons, electrons are a negative charge, think negative numbers are smaller? Whatever works to help you remember, reduction is addition of electrons, not removal. And again, it's the electron and hydrogen proton exchange that's more significant than the oxygen exchange. Now these things are linked. Anytime you have an oxidation reaction, you have to have a reduction reaction going along with it. In other words, something is oxidized, something else is reduced. Consequently, you'll hear them referred to as oxidation reduction reactions. They're both happening at the same time. Or sometimes in chemistry they call them redox reactions, because people get tired of writing out oxidation reduction, and it's harder to say reduction oxidation, I guess, so they just say redox. However, if I ask you what kind of a reaction involves the addition of oxygen to or the removal of electrons and hydrogen protons from, don't tell me that's a redox reaction, because I'm going to tell you that's only half right. You have to separate them. You have to know which is which. Redox is both reduction and oxidation. Now, another thing that's really significant when we are looking at coenzymes, the NAD and FAD, NADP, when they pick up electrons and hydrogen protons, they are reduced. In other words, they've undergone reduction, but they are higher in energy. They're holding on to those electrons, and that gives them a higher energy potential. So NAD and FAD are the oxidized forms. NADH plus H plus and FADH2 are the reduced forms, and that's a higher energy potential. This is significant when we start looking at metabolic pathways. And again, these things are always linked. When oxidation is happening, reduction is also happening. Something's going to give up the electrons. Where do they go? They're going to get on something else. The molecule giving up the electrons is oxidized. The one gaining the electrons is reduced. Consequently, you need to know the difference. So there's a mnemonic device. There's more than one, but I like Leo the Lion Goes Grr. You can remember Leo the Lion Goes Grr. LEO is lose electrons, be oxidized. Grr is gain electrons, be reduced. And you can look at a growling lion. Grr. Lose electrons, that's oxidation. Gain electrons, that's reduction. Grr, grr, Leo the Lion Goes Grr. You could also use oil rig. Oil rig is oxidation is loss, reduction is gain. I don't like oil rigs. They burn up. So go with Leo the Lion Goes Grr. Okay, another way to categorize enzymes is constitutive versus repressible versus inducible. This has to do with regulation of enzyme production. Enzymes are proteins. Where are proteins made? At the ribosome. But to get to that point, the cell has to make RNA, messenger RNA, also has to make transfer RNA and ribosomal RNA. But anyway, then the message has to go to the ribosome and the protein has to be built. Well, this can be regulated at different levels. We're going to talk about repression and induction after we go through genetics, because if we were to talk about it right now, it probably wouldn't make much sense. But, yeah, it has to do with their production. If enzymes are constitutive, the term constitutive refers to enzymes that are absolutely required by the cell. The cell has to have them. Consequently, their production can't be turned off. They can't be inhibited, or the word repressed. They can't be repressed at the gene level, can't repress the production of constitutive enzymes. They're always being made. It turns out most enzymes in the cell are constitutive. And then if you think about, well, what kind of enzymes do cells have to have? We've just been talking about making ATP. So ATP synthase, that's a constitutive enzyme. The cell has to have it because they're constantly making ATP. If they're going to replicate their DNA, they have to have DNA polymerase. If they're going to make RNA, they have to have RNA polymerase. All of those are constitutive enzymes. So ATP synthase, DNA polymerase, these are good examples of constitutive enzymes. But again, most enzymes within cells are constitutive. There's only some that their production can be regulated at the gene level. And we'll talk about inducible and repressible later. Repressible enzymes, yeah, repressible. All it means is that the genes that encode those can be turned off. And it's not the gene that's turned off, it's transcription. If they're inducible, transcription can be turned on. And never mind right now. We'll go over it later. Okay, so we have enzymes. They're proteins. Now we can talk about what kind of factors influence enzyme activity, in addition to coenzymes, cofactors, prosthetic groups. So there's other things that influence enzyme activity, and some of these you can relate to because of other topics we've covered. For example, temperature preferences, psychrophiles, mesophiles, thermophiles, hyperthermophiles. Why do you suppose organisms grow best at certain temperatures? Because their enzymes are going to be most active at certain temperatures. So temperature is a factor that influences enzyme activity. Again, heat would tend to increase molecular activity, more banging of molecules into one another. But heat influences whether enzymes are going to function or not. So if you have psychrophiles, their enzymes are going to work best in the cold. Something about them, their configuration, makes them work best in the cold. If the cells are thermophiles, their enzymes work best in the heat. I think I mentioned the DNA polymerase that comes from Thermus aquaticus works best at 72 degrees centigrade. That is hot. That enzyme works best at very high temperatures. Of course, if you get enzymes too hot, they're going to be denatured, and that just means they're going to lose the configuration that is required for them to do their job. They have to have a specific configuration in order to bind with the substrate. If they're denatured, they lose that. So if you get them too hot, they're denatured, and then they don't work anymore. Another factor that influences enzyme activity is pH. So we said organisms tend to have a specific pH at which they work best. So we know that we can change the environment either by causing it to be more acidic or causing it to be more alkaline. If you change it very much, enzymes again will change their configuration and then they don't work properly. That's why most organisms prefer to have an environment that's fairly neutral, around 7. But we know that there are organisms that are thermoacidophiles. Their enzymes work best in acidic environments and also hot environments. The enzymes that can do that are used in industrial processes, so people are interested in them. Another factor that influences enzyme activity is concentration. This can be concentration of the enzyme or concentration of the substrate. Both are going to have an effect. So in this diagrammatic representation, you can see that as you increase substrate concentration, activity of enzyme goes up. If, let's say you have a set amount of enzyme, as you increase substrate concentration, the activity goes up until you max out your enzyme. You've saturated the enzyme. You can't have any more reaction. Another analogy, imagine that I have a pile of sand on this side of the room, and my reaction that I want to catalyze is moving the sand from this side of the room to that side of the room. And my enzyme is a bucket. If I have only one bucket, I can only move the sand one bucket at a time. The bucket represents the enzyme concentration is the limiting factor. If I could somehow magically move 10 buckets at a time, I could move the sand 10 times faster. Enzyme concentration goes up, reaction rate increases. But if I had 20 buckets, and there's only one bucket worth of sand over here, my reaction is still going to go very slowly because the substrate level, the concentration of substrate is going to limit my reactions. So it can either be concentration of enzyme or concentration of substrate. They'll both influence reaction rate. Another factor that can influence enzyme activity is light. This is one that you don't see this much in biology textbooks, but we know it's true here because there are specific enzymes associated with bacteria that are light activated, activated by visible light. So if they're not in the light, they don't work. These are involved in DNA repair. So there are enzymes that are light activated. It can be another factor. And then there are inhibitors and enhancers. So we don't spend a lot of time talking about enhancers, which are also called activators, but I do want to spend a little bit more time talking about inhibitors. There's two categories of inhibition. We're going to talk about enzyme inhibition. The first category is called competitive inhibition. If an enzyme is being influenced by competitive inhibition, what that means is that something, the inhibitor, can compete with the normal substrate for the enzyme active site. The enzyme would normally bind a certain molecule. The inhibitor is something that looks enough like that molecule that it can get into the active site instead. So the competitive inhibitor is something that can get into the active site of the enzyme in place of the normal substrate. And then it's going to block that enzyme. Here we see an example of this in a diagrammatic form. Here's our enzyme. It has this active site. Here's our substrate that would fit in there, but when this inhibitor is in place, the substrate cannot bind. So that enzyme cannot pick up its substrate. It can't do what it's supposed to be doing. This type of inhibition can be influenced by, again, concentration of the inhibitor. If you have only a little bit of inhibitor, it's not going to be doing too much damage. But if you have a lot of it, you can block all of the enzyme active site and your enzyme can't do anything. This is significant because there are a number of metabolic toxins that are competitive inhibitors. So toxins such as arsenic, cyanide, and nerve gas, or sarin. These are competitive inhibitors of specific enzymes. When those enzymes are bound by the inhibitors, they can't carry out their function. So why is cyanide such a significant toxin? Because it binds with a molecule at the end of your electron transport chain. Normally at the end of the electron transport chains, electrons have to be transferred to oxygen. Cyanide blocks that. If you can't transfer electrons to oxygen, then you can't run your electron transport chain. You're basically stopping it by blocking the last reaction. And cyanide will kill you. Nasty stuff. The other form of inhibition is called allosteric inhibition. And allo means other. So what this refers to is an inhibitor that's going to bind to a site other than the active site. It's not competing with the normal substrate for the active site. It's binding someplace else. When it binds to the protein, the enzyme, it causes a confrontational change in the protein, and then the active site doesn't work anymore. So in this case, here's our enzyme. The active site can bind something that has a nice big circular shape unless our allosteric inhibitor binds. When the allosteric inhibitor goes into this site, it changes the active site and now the substrate can't bind. That's allosteric inhibition. That's a really simplified version. This is showing the same kind of thing. Sometimes you'll see this called non-competitive, but we're going to call it allosteric because the inhibitor is binding to an other site, a site other than the active site, and that's significant. You can get on Wikipedia and read about allosteric regulation. You're going to find out there's a lot more than just allosteric inhibition. So that's the only thing we talk about in here. But there's allosteric regulation that can be either inhibition or it can be activation or it can be enhancement. There's a lot to it. So we're just looking at allosteric inhibition. If it was activation, again, it's allosteric, so that a compound can bind to an enzyme at some other location and change the active site so it can bind something that it couldn't bind previously. So it can enhance the activity of the enzyme to do a specific thing. And the other thing about allosteric regulation is it doesn't just apply to enzymes. There's a lot of proteins that aren't enzymes, and apparently lots of different types of proteins can be subject to allosteric regulation, which we're not going to worry about. The last thing has to do with enzyme names. We're going to encounter a number of enzymes, and you need to learn the names of some of them. Well, actually, you have already learned the names of some enzymes. Enzyme names often end in ASE, so if you have a term you have to define, and it ends in ASE, at least write down, this is an enzyme, and then try to figure out what does the rest of the word tell you about the enzyme. Because it'll often tell you either what the enzyme acts on, or what the enzyme does, or it has something that relates. So for example, you've seen an enzyme, well, which enzymes have you learned so far? So luciferase is a good example. Luciferase, that's the enzyme associated with bioluminescence. So luciferase, notice the ASE ending, and then you think Lucifer, what does that have to do with? Oh yeah, Lucifer was either the angel of light or the light bringer or whatever, it has to do with light. So luciferase is an enzyme associated with bioluminescence. How about ATP synthase? There again, ASE ending. If it's ATP synthase, it tells you what it does. It synthesizes ATP. So the name, it ends in ASE, and it typically tells you something about the enzyme. In this case, something that synthesizes ATP is ATP synthase. A polymerase. A polymerase is an enzyme that catalyzes the formation of a polymer. We're going to talk about DNA polymerase and RNA polymerase. They build DNA or RNA. So they're enzymes involved in making polymers. So there's a bunch of them you get to know. I was going to say, we talked about hydrolase enzymes, but hydrolysis, that's very generic. Hydrolase enzymes are actually proteinases and lipases and so forth. There's different categories. But you're going to get to know some other types of enzymes. If I said there was an enzyme whose name was aminoacyl tRNA synthetase, what do you suppose that one does? Aminoacyl tRNA synthetase. It puts together an amino acid and a transfer RNA. Again, the enzyme names often tell you what they do. So try to pay attention to that. That's the end of that presentation, so I'm going to stop. That's probably good. Because you guys have a quiz. So we'll have our quiz at 7. Before that, maybe I'll give you a test.
Well,
that's why it says 1 billion. And then it's 10 to the 9th, a lot of them in there. Copy paste it and just put it in there. And just figure it out. So there's 1 billion. Or 10 to the 9th. Damn it.
after I just like memorized that. Maybe short.
Like that, probably. So if you don't remember, we'll try to be fine. I just made my mind too many times. But you'll remember. Because I know you're probably like, so we'll help you remember.
10 to the 9. I'm just gonna write it right here.
Yeah. I
That get down.
I can refresh.
Oh, I know, me too.
I mean, down a little bit of coffee real quick.
Remember that tonight's quiz is just one lecture worth of information. Microbial nutrition and growth, right?
With 20 questions still, right?
Yeah.
20 points.
I had a hard time making it 20, but I managed.
Okay.
I had to come up with some innovative questions.
Oh boy.
Oh boy.
Good.
Well, some of it was
How many-
review, the things that influence growth. These are the soil. These are the soil. Is this your morphological unknown? I'm gonna put it in this bag. These are clean plates. These not. Yeah, these got fungus in them. They're not pure cultures. They're just, they were from our dirt. But I do need to know that you're putting them in there so that I can keep track of it. Yeah.
Is it just like 3 for the soil?
Yeah.
I was like, there's not 4.
No,
There's 3.
There's 3. There's Azotobacter.
Yes.
Pseudomonas, Pseudomonas, and
Yes.
Yes.
yeah. So if you're turning those in,
Oh, well, I'm doing like
It's the 8.
yeah. So I'm just gonna make a note that you turn these in or some back someplace.
yeah, I was pretty good.
Yeah. I was thinking I had some that had already been used, but that's okay. We'll put it in.
And then the fungus, should I put all my, because I've got illustrations, but I'll have them still together.
Oh, I'll stick them together. Yeah.
So I have that one, and then I've got my more unknown one.
Now that plate you just put in here.
Yes. And then I've got the fungus. Sorry.
No, that's fine.
I've got it all.
We just want to get it all coordinated.
Yes.
Yeah, I just want to make a note on here that the plate is not fitting in the spot.
It's the 8.
Yeah, that just.
Tetron is the 4.
Okay, thank you.
Tetron is if the plane changes once.
Ah, heartbreaking story.
Did you do better?
I did a lot better, but like literally 0.7 away from. Literally like I get such bad 0. I got a 7.3. I get such bad test anxiety.
I do too. I do too.
I was so heartbroken. I was like, whatever. I just started getting my alcohol.
Well, which one are you trying to get? We're trying to get all the Sierras. Chico's? Oh, so yeah. See, I just barely.
I'm like, it's fine. It's fine.
Like sometimes I really want that.
Study take it. And like,
But you still did what, like 5% or something like that?
yeah, I did like,
That's
I
more
did
harder. I need like 7%.
I did better, and I was like, okay, well, I guess in that way I'll just do like I do, keep going, and it's gonna be like this for now.
Well, I mean, for Chico, you can take it minutes.
You can. So I'm like not stressing out that much about it. And I looked at it because I got a 3.6 overall GPA for my sciences and for everything else, and I'm like,
I'll put my hands up like you're
Oh, tax day now. Nobody fucking gets I got an 80 and I have a 4.0 nursing GPA and I can't get it. Like, there's no way to get it.
Just really stressing out. I was so focused on this. Like, I did okay.
Yeah.
So I got all my homework done and I got all turned in and I'm like, it's all done. The only thing I have left to do is my extra credits.
The population, yeah, I started doing it.
I was gonna do it and I was like, oh, I'm gonna get everything. And I did, and that's a relief because now it's not next month
So
coming up.
there is a study guide. That's posted. I think
There's always so much work.
it's
Like as soon as I'm done, I'm like,
so you have all these different
you're not
things,
gonna
and then there was a study guide.
I know. But I'm fine, and like having to study for both at the same time, like I was gonna like have a heart attack. So I'm glad I don't have
So
to do
it'll
that.
show you
I can
all
just
taxonomy.
It's like if you don't have to work and you don't have to do other like
I work
the extra
full-time.
things, yeah, exactly. Then it's like that. It's gonna be
Take the
different
pictures off the website.
when you don't have to do other
Right when you're not
extra
working.
shit. Yeah.
On the back side.
Yeah.
What do you have to
What did you do
know?
on this exam?
If it's
I got a
fungus, you have to
75,
do sexual
which all
force,
things
station,
considered, I was like,
and then
I
the
was like
taxonomy.
so
Which is
focused on
domain,
these. I was
carry,
like, you know
And
what?
testing is like as big of a
It didn't complicate as much as I thought it would, and with everything I just turned it on, like it's just gonna go right back up. As long as I get a B, that's seriously at this point, I'm like, and then you can be done.
Yeah, she's gonna upload
The study guide
three homework
just,
assignments by Thursday.
people get really upset with me.
Oh,
and say,
really?
"You asked exactly the same question. I won.
I got it all
These are
turned in, I was like, I don't
It's
know
to
about
get you to think
don't worry
about
about this, and I was like, then,
You
turned it in. Yep, I turned it
And
in.
there's,
So
if you look at the study
yeah,
guide online,
fungus
there's
homework.
two examples.
And Chico only takes your 2 highest science
Those are very
grades.
important to
Oh,
study
yeah.
because
Really?
it tells you
Yeah.
Oh, it's a good thing I've gotten.
composition of the whole thing.
Huh?
But
Yeah,
are
no,
those the only questions I should ask? No.
Really? That's kind of cool.
In other words, I try to make up new
You know,
questions.
Done a couple of them.
You know,
Last semester I got a B, and I was like,
Exactly. I killed
I will
someone.
take that.
It's drove me crazy.
Yeah.
I was just like, I'm gonna kill her. Like, we didn't get out of that class soon. I was just like, I'm gonna murder her. Not as much as Sonia wanted to, but like,
Surviving
You probably have a lot of
was, like,
points
the
for
best
Chico,
for me.
honestly. And you're fluent in a language.
Because I was so done.
yeah, I was just done with her. I was
That's
just like,
what
no. And I almost
I
didn't
had
get
my
into this
teacher,
class because I was
I
on the
had
wait
my
list
Spanish
at
teacher,
first.
I emailed her
And
like,
so I
I was
enrolled
in
her.
yesterday when it was due, I was like,
waitlist, and then as
can you
soon
sign this
as
for me?
I
Like,
was
just tell tell Chico that I'm like proficient in Spanish.
Because she
Yeah,
was the
sure,
first
no
test grade
problem.
at
You want
first.
Sent
the
me
last
back.
I remember
I was
that.
like,
Did you email her too?
I like
I-
cried. No, I just
Proficient
cried.
in
I was
Spanish.
like, oh yeah.
And
Me,
so
I
then-
emailed her,
and I was
Sonia
like,
emailed
Because
her,
if she
Diana
hadn't
emailed
changed
her.
it, I would have gotten a C. I was
Yeah.
ready for that quote, and I got it.
Yeah. Because she gave us all like it said I got like a 70% on it, and I was like, I
It said I got a 65 on it.
exactly, and I got like in the 90s.
And then I got it.
Yeah, exactly. So it was like 10% like off.
I was like, that's
Because I don't want
crazy.
to take the
I'm gonna apply in Fresno.
I like lost it, and I was like so sad, and then I like saw the email, and I was like,
Oh, and
So you
Diana
high for
emailed her about it, like, you know, like, oh, blah, 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 like, what's up with this? And then she literally emailed her back and was like, if you have a problem with your grade, I'll leave your grade in the office and you can go in on your off time to go look at it. Like, she wasn't even gonna say anything, so then we all emailed her because she texted us and was like, look what she sent to me, like, is your guys' grade like that? And I was like, yeah, mine definitely is the same grades. Then we all emailed her, and then she finally looked, she's like, oh, whoops, and I'm like,
because she puts out a 125, not 100. So she was putting it out of 125, like our grade out of 125, not 100. It was out of 125.
oh, I had no
And
idea.
she like put in our score out of 125.
Sac State was
So
like,
that's what happened.
well,
I was like, fucked
up.
See, I had no idea what she did, but I was just like, I know this isn't right because I know I did not do.
I know, I was like, I did that bad. I studied so hard.
Yeah.
Oh my gosh.
And I was like, I came out of that like okay.
I
know I
felt
did.
okay.
Yeah, I felt like okay about like I felt better about that than some of the other tests which I did better on. So I was like, I know I did not get below those. It's like I was done with her though. By the end, I was just no, and she didn't want me here in her class either, so it worked out for everyone.
She wanted like,
She didn't want any of us in her class. She hated her class.
and she was freaking out about my program. She was like, it's gonna be so intense and
I
all
know.
this stuff.
She
I'm like,
said that shit all the
no.
time.
And no, I'm
Yeah,
like,
you're just like, don't put that evil
I
in me right
barely
now.
survived that class.
Exactly. Like,
But
don't
then
put
I've
that
heard
evil
she's
on.
super chill. I'm like chiller than physio.
Oh, who Bird was chill?
Because she was super crazy about anatomy, which I heard, and then physio was like, oh my gosh, and everyone's like, oh, she's so nice about micro, it's her favorite class. I'm like,
Yeah,
she was
exactly.
not like that.
That's what people say about Cardozo. They say she's so much better than micro because she likes that subject.
That's how Bird was too, because she'd always talk about micro, and then we'd be like, okay, well, we're in physio right now, so we're gonna do this. be here.
She would always try to incorporate OChem into whatever we were learning. And we're just like, well,
And you're just like, calm down, this isn't true.
yeah. I never stopped it. I never stopped